Okay, so we are going to start with medical mycology, which is nothing but the study of medically important fungi. So let's get started. So, fungi is, as we all know, a eukaryote. Okay, and fungi are obviously non-motile organisms, and they can be either unicellular or multicellular. Okay, we will get to know about them: unicellular like yeast, okay, and multicellular like other filamentous fungi. Now, the fungus have a peculiar cell membrane because it contains the cell membrane of fungus or the fungi are going to contain ergosterol. And this ergosterol is kind of special because this is the target of many antifungal drugs such as amphotericin B or imidazoles. Okay, then what is fungus cell wall? Okay, the fungus cell wall is made up of chitin, which is a homopolymer of Naga. Okay, and Naga is N-acetyl glucosamine. Okay, now we are going to see the classes of fungi. So fungi is divided in 4 classes. Okay, so first we have zygomycetes, which is also known as phycomycetes. Okay, then we have ascomycetes, basidiomycetes, and deuteromycetes. Okay, and the sexual spores that I am going to tell now are the only ones that we have to remember in the entire mycology. So that is good because there are very few. So we have zygospores or oospores and oospores in case of zygomycetes or phycomycetes. In case of ascospores, we have ascomycetes, we have ascospores, and we have basidiospores in case of basidiomycetes. Okay, but in case of deuteromycetes, we have none. We have no sexual spores because the sexual state of this class of fungi has not been discovered yet. Hence, it is also known as fungi imperfecti. Okay, it is imperfect because its sexual state is not known yet. Alright, so now we are going to see the various morphology of fungi. Okay, so morphological classification of fungi. Let's get started. First, we have yeast. Okay, yeast or true yeast, and these are the unicellular fungi I was talking about. These are unicellular fungi. Okay, and they reproduce. They are going to reproduce asexually by budding. Okay, in budding, what happens is, let's say this is your parent cell, and it is going to form a cellular protuberance which is going to grow. Okay, it is going to grow and gets detached, and we have two yeast. All right, so we have to know some examples. So we have a pathogenic and non-pathogenic yeast. So the non-pathogenic one is your Saccharomyces cerevisiae, and the pathogenic yeast is Cryptococcus neoformans. Okay, it is an encapsulated yeast, and we use negative staining, that is we use India ink or nigrosin for its microscopy. Okay, so it is encapsulated. Then the next one we have yeast like fungi. Okay, the next morphology. So these are the fungi which are again going to reproduce by budding, but this is different from what we saw earlier. They are going to form that cellular protuberance, but it is not and it is going to grow, but it is not going to get detached from its parent cells. So they are just going to grow and form more chains. Okay, so this whole chain that I grew, this is known as a pseudohyphae. Okay, so this is a pseudohyphae. Hyphae is actually a cylindrical cell which we are going to see in filamentous fungi. And in case of yeast like fungi, we see these pseudohyphaes which appear like hyphae, but they are not actually hyphae because at the junction of these parent cells, that is at these septa, they have constrictions, right? The normal hyphae appear somewhat like this, the septate one. Okay, they have no constriction, but in case of yeast like fungi, we are going to see constriction which represent your pseudohyphae. Okay, so they have pseudohyphae, And we have just need to we just have to remember just one genus which have this yeast like fungi, that is your Candida. So Candida की जितनी सारी species होंगी, they have this yeast like fungus morphology, they have pseudo hyphae, okay, except one species, okay, one species which will not produce these pseudo hyphae, and that species name is Candida glabrata. Okay, so this is important. Candida glabrata is the only species of Candida which does not have yeast-like fungi, which does not have yeast-like fungi morphology. Okay, then let's get to the next one. We have the filamentous or the filamentous fungi or mold, which we call mold. Okay, so they are made up of the cylindrical cells that we were calling hyphae. Okay, the cylindrical cells known as hyphae, and hyphae can be septate, okay, that is partitions, okay, so they can be septate or aseptate. The aseptate hyphae are found in only one class of fungi, that is your zygomycetes or phycomycetes. Okay, so that is good news, just one class. And septate hyphae you will encounter in the fungus like penicillium and your aspergillus, and obviously your dermatophytes, that is trichosporum, microsporum, epidermophyton. Okay, so let's see the morphology here in this diagram. Okay, so this is the yeast cell, okay, the parent cell, which is going to form this cellular protuberance, and which is going to grow and then detach and form again another cell by reproducing like budding. Then we have these pseudohyphae, okay, they have these constrictions and they are not getting detached like in case of yeast. And again we have these true hyphae or the hyphae, okay, this is the septate hyphae and this is aseptate. And yes, what is mycelium? Mycelium is nothing but when a bunch of hyphae is present, we call it mycelium. Okay, so now the fourth morphological form that we see in case of fungi are thermally dimorphic fungi. So what are thermally dimorphic fungi? Thermally means temperature, okay, something to do with temperature. Dimorphic means to morphology. Okay, so what do we mean by this? That at two different temperature, two different morphological forms are seen. Okay, so thermally dimorphic fungi में क्या होता है? At your body temperature, that is at 37 degrees Celsius, and at the environmental temperature, that is 25 degrees Celsius. Okay, so at 25 degrees Celsius, that is in the environment, when the fungi is not in your body or in any human's body, and there is no infection yet, so it is present in its infectious form. Okay, that is what its infective form is. And how does it appear at 25 degrees Celsius? It is going to appear like a filamentous fungi. I mean, it will be a filamentous fungi. Okay, but when you get infected by this thermally dimorphic fungi, it is going to change its shape. Okay, it is going to change its morphology, and it will become, okay, it will look like a yeast-like morphology. Okay, so this actually I saw in my microbiome lab. The technician asked me that they had these two culture tubes which had the culture medium SDA, which we will talk about later. Okay, so they asked me, okay, why we use two culture tubes for this patient sample. So for the same sample they were using two tubes. So what could be the reason? Because they are going to incubate it at two different temperatures, that is 37 degrees Celsius and 25 degrees Celsius, to confirm whether it's a thermally dimorphic fungi or not. Okay, that is one way, and we are going to see other ways to diagnose it. Anyways, the names of these fungi are not so cute, so we have this mnemonic for remembering their names. So let's see what is the mnemonic. Okay, the mnemonic is his pistol shoots blank checks. Like, wow. Okay, so only if. So we have his pistol shoots blank checks. So from H we have Histoplasma capsulatum, from P we have Penicillium marnife, from S we have Sporothrix shankai, and from B we have Blastomyces dermatidis, and from C we have coccidioids immitis, or and not or, and we have paracoccidioids brasilensis. All right. So these are the only thermally dimorphic fungi that we need to remember. Okay, so that was about the morphological classification of fungi. Now we are going to see how are we going to diagnose a fungal infection, right? So when a sample in your microbial lab comes, then how are you going to detect like which fungus is it? Okay, so we have various methods which we are going to see one by one. So first we are going to do a KOH mount. For example, you get a specimen sample, like you get a skin scraping. Okay, so specimen like skin, your hair, or not your hair, but anyone, the patient's hair. Okay, the skin, hair, or nail. Okay, so these are the samples for which we want to do a KOH mount first. Okay, and then we will look under the microscope. Why are we doing KOH mount for these specimen? Because these specimens have keratin. Okay, and what is KOH? Potassium hydroxide work as work as a keratinolytic agent. Okay, so it is going to literally eat up the keratin in your hair and the skin and the nails. Okay, so we use a concentration. Okay, we use a concentration of 10% KOH in case of skin and hair, and in case we have the nail as a sample, then we want to use 20 to 40% concentration of KOH. Okay, now this is how it's going to look under the microscope after you have done the KOH mount. Okay, these are the pseudo hyphae actually. Okay, these are pseudo hyphae of Candida albicans. I got it from Google obviously, but and that's what it was written. I'm not sure though. But obviously this is Candida albicans because we can see the pseudo hyphae and we know the genus Candida does have these like fungi. That means they are going to have pseudo hyphae. Okay, then what else you can do? You can do gram staining. See, all the fungi are gram positive only, but we have the filamentous fungi. Their hyphae going to stain very faint. Okay, so they do not take up the stain that well, but eventually they are also gram positive. So we use gram stain for yeast or yeast-like fungi. Okay, which is very understandable. Then we have the India ink or nigrosin, which I was telling that we use in case of Cryptococcus neoformans because it is used in negative staining. Okay, negative staining to see the encapsulated organism, and Cryptococcus is one of them. Cryptococcus neoformans. All right, then we have some very weird named stains. Okay, the fungal tissue stains. I do not like their names at all, but we got to learn. First one, the recommended stain for the histopathological staining of the fungus infected tissue, we have the PAS stain. Okay, that is periodic acid Schiff stain. Okay, this is the recommended one, and it is going to stain your fungi dark pink. Okay, dark pink. And for you to stain the fungus with the PAS, the fungus have to be alive, so it stains live fungus. Okay, that is not the case with the GMS stains. Okay, that stain both the live and the dead fungus. Okay, so what I am talking about is GMS stain, that is gomor methenamine stain. Okay, it will stain both the dead and live fungus, and the fungus is going to appear black. Okay, the fungus is going to appear black in color, and the background will be somewhat pale green. Okay, then we have some other stains like mucicarmine stain. We use this mucicarmine stain for two fungus specifically, that is Cryptococcus and Rhinosporidium. Okay, then my favorite one is the my favorite one is calcofluor white stain. because it is going to show this fluorescence that we are seeing right here. So calco fluor white stain. Okay, so it is more sensitive than the other stain that I talked about. It is more sensitive, and the good thing about is that you will stain the chitin. It stains the chitin of the cell wall, okay, which was the homopolymer of NAGA, N-acetyl glucosamine. So this is a picture of calcofluor stained fungi, and it sort of appear like a septate hyphae. Okay, let's move on to the good part. Okay, so we have done till now KOH mount, we have done gram staining, we have done the histopathological tissue stains for the fungus, and we have done, in case of encapsulated organisms such as Cryptococcus neoformans, we have done the negative staining, that is by using India ink or nigrosin. Now we are going to do fungal cultures. Okay, and the most common medium for fungus is SDA. Okay, sabrods, dextrose, agar. Okay, so this is the most common medium that mostly the fungus grow on. Okay, most of the fungi will easily grow in this SDA medium, and you have to remember, okay, the pH of SDA is 5.6. Okay, and then what other culture medium we have in our options? Okay, there is one neutral SDA. Okay, we're calling it neutral because the pH is 7.2. Okay, and there is just slight difference in neutral SDA and SDA because of the concentration of peptone, water, and dextrose and all. Okay, then we have what is known as chrome agar, and it is actually very cool. I saw it in the microbiome lab, and it is okay. It is basically for Canada. Okay, and different species of Candida are going to give different color on this medium. Okay, so this is the culture plate on which chrome agar medium will be there, and we will inoculate. We will put the we will culture the Candida here, and they are going to appear very nicely with different color. Okay, so that is why it is known as chrome agar. Okay, मैं तो same color से बना रही हूँ, but same color नहीं होता. It different colors होते हैं. Anyways, so okay, so we can do the sabrodextrose agar, we can do the culture from this SDA, or we can use chrome agar for Canada, and we can use neutral SDA as well in some cases. And then there comes one other medium, which is BHI agar. What is BHI? It is brain heart infusion agar. Okay, so why do we use this brain heart infusion agar? It is because it is an enriched medium. Okay, it has additional nutrients or things that a fungus can need. So normally your fungus is easily going to grow on SDA, but there are always some high maintenance people. Okay, some high maintenance fungi are there, which are the fastidious fungi. Okay, fastidious means these high maintenance people, they need more things. Okay, so they will grow on this BHI agar. Okay, so that is about the diagnosis, various methods of diagnosis for the fungal infection. Now we are going to see the classification of fungal diseases. Alright, so we have classified fungal diseases in 4 categories. We have superficial mycosis. Mycosis means fungal infection. We have subcutaneous mycosis. We have systemic mycosis. And we have opportunistic mycosis. Alright, superficial mycosis, सुन के ही लगता है that superficial things are going to be involved. So what superficial things are going to involve in this infection is your skin, your hair, your nail, alright, for which you want to do your KOH mount. Yes. Then in subcutaneous mycosis, again, all these can be involved, plus your subcutaneous tissue, and plus minus the bone involvement could be there. Not in all cases, but in some cases. Systemic mycosis also we are going to see in very much detail. What are opportunistic mycosis? See, opportunistic mycosis is the one which normally do not infect the immunocompetent people. You know, the people who have very good immunity, okay, they are just fine. But they are going to infect those people which are immunocompromised, such as your HIV patient, or someone who has gone under organ transplant, okay, or someone who was taking a lot of steroids, okay, corticosteroids. So these people have low immunity, and this will lead to infection. This will lead to some opportunistic infections, and some of those could be your opportunistic mycosis. Okay, so that's about the introduction of medical mycology.